Hello everybody. Today we'll talk about the taste perception. Taste system is the sensory system that is responsible for the perception of taste. So the stimulant or taste stimulus is called testants. Okay, so testant is the stimulus or the signal. Okay, the molecules signal molecules that binds with the receptors, okay, and give the perception of receptors. Okay, so testant are the test molecules and that must be able to bind with the receptors in the tongue, and that would give you the perception of taste. So taste and molecules must be recognized by the receptors, taste receptors. And the taste receptors then will do what? Produce the neural signal. Okay. So testants must be able to bind with the receptors and produce neural signals that the brain represents as the perception of different tastes. So this neural signal goes to the brain, and brain gives you the perception of different tastes. Okay. Okay. So perception of taste. Now, the taste and flavor are two different things. Okay. Flavor is more complex mechanism in the brain, and the flavor perception is more complex, okay, and multidimensional than the taste perception. So flavor is more complex and multi-directional. A number of factors are involved uh, in flavor. So flavor gives you the enjoyment, okay, of taste. So several factors are involved. When you eat the food, you know that a number of things give you the enjoyment of the food. For example, when you are eating, the smell, of course, the most important one. Without smelling the food, you don't enjoy the food. However, there are several other factors that can uh, uh, improve the flavor or enjoyment of the food. Okay. Now, why we need to taste? We need to eat the food, right? uh, and the food we eat contains very important chemicals, nutrients, right? Proteins, carbohydrates, fats, those are essential for your survival. You have vitamins in the food, right? You have minerals. So all these are essential for your life, as well as water. Okay. Now these chemicals inside your body are used. The nutrients are used to produce ATP, right? That gives energy. So you need to eat, right? And synthesis of protein. So synthesis reactions or process is needed for repair the tissue repair. You know that injury occurs in the tissue inside the body. We don't see from outside most of the injuries that occur inside the body. Cells are dying all the time, and new tissue, new cells are being produced, right, to replace the dead ones. So that is the repair, and you need the food nutrients for that. Okay, for chemical reactions, you need these chemicals for the chemical reactions required uh, to synthesize the hormones or enzymes or other chemicals. So taste is very important, because if you don't taste the food, you won't feel the urge to won't feel the uh, urge to eat. So for energy, for repairing, and also for maintaining the chemical balance, you need to eat. Now, 5 tastes are considered as basic tastes. 5 basic tastes. Before, 4 tastes used to considered as basic tastes. Now we have 5, because protein, taste of protein, that is called umami, has been added later. So what are those 5? Sweet, sour, okay, then uh, umami, bitter, okay, and salty. So these are 5 basic tastes. Uh, there is another one, that is the taste of fat or fatty acids. That is not considered as a basic uh, taste. And umami, this one was first introduced by a Japanese scientist, a taste that evoked by testants such as amino acids, that means protein. Okay, so those are 5 basic tastes. So the substances that give these taste perceptions, let me quickly just go over. Sweet taste is given by commonly sucrose, fructose, glucose. So glucose, fructose, sucrose. Glucose, fructose, these are monosaccharides, and then sucrose. So these are the main chemicals or substances that give the sweet taste perception. Salty is given by sodium chloride, which is table salt, the most common one, potassium chloride, lithium chloride. So all end with chloride. Okay, so those are the chemicals give the salt or salty perception. Then umami, as I mentioned, the amino acids. And amino acids, there are many different types of amino acids. Monosodium glutamate. So glutamate is the common one. Probably you have heard. Glutamate. So glutamate is one. Arginine. So those are common chemicals for umami or protein taste. And bitter is given by the bitter taste is very important because it gives you the signal of toxic chemicals. So usually the toxic chemicals are bitter tastes, and we can quickly detect that and avoid that. So that is one type of protection that you particularly just think that we have many ways to select the food, but many creatures, they eat live in the forest and eat without much uh, cognitive functions. So they just need to detect after they put the food inside the mouth. We can think which one we should eat or not before we put them out. But so that taste that gives them the signal that it should be immediately avoided after putting in the mouth. That's the bitter. And quinine, example, caffeine. Those are the chemicals give bitter taste in human tongue. Okay, so then the sour. Sour is given by acid. So citric acid, is sour. Acetic acid, hydrochloric acid. So all acids, acidic taste. Okay, so citric acid, acetic acid, hydrochloric acid, those are good examples of the tastants or the chemicals that give the sour. Okay, so then uh, The peripheral organ for the taste or gustation is the tongue, and tongue gives the perception of taste. You know that this is the peripheral organ, but it has many other functions like production of speech, digestion of food, mixing the food with saliva, and swallowing of the food, all those functions. But one important function of the tongue is tongue has the receptors for taste, and there are four different types of taste, structures that contain the taste receptors or taste buds. Those are called papilla. So there are four types of papilla that you can see actually on the surface of the tongue from outside. And among those four different types of papilla, three of them have taste receptor cells inside, and one doesn't have the taste receptor cells. So which three papilla have taste receptor cells inside, or taste buds inside, are fungiform papilla, foliate papilla, and circumvallate papilla. Filiform papilla are empty. Taste buds are not present inside. Okay, so there is no taste receptors in it. Okay. Now the papilla are distributed throughout the tongue, and we'll see first the location of three different types of papilla, where they are located. Okay, so these you see on the tongue, these tiny structures, they have different shapes. These are the papilla, three different types of papilla. So these are tiny structures on the surface on the tongue. Most of the papilla are on the surface of the tongue. However, taste receptors are also present outside of the tongue in smaller number. For example, in the pharynx or epiglottis. However, mainly on the tongue. Okay. First, let's see the fungiform papilla, tiny mushroom-shaped structures located along the edges and top of the front two-thirds of the tongue. So, if you see, these are the fungiform papilla, and most of them, almost all of them, are present in the anterior two-third part of the tongue. Some are along the edge. Here you see, not in the center, and many are in the tip area, or most front part of the tongue. And each, if you open a fungiform papilla, each fungiform papilla contains 3 to 5 taste buds. So each has 3 to 5 taste buds. Okay, and then if you see the back part of the tongue, in both sides you have the foliate papilla. They're like folds. the posterior and lateral, both sides of the tongue, and they are like ridge-like folds of tissue located on the sides of the tongue near the back. And in each fold you have about a few hundred taste buds. Okay, so the number of foliate papilla, although is less, much less that fungiform, however inside each fold or papilla you have hundreds of uh, taste buds. Okay, few hundreds, 100 taste buds. And then in the dorsal back of the tongue, the upper surface, you have larger, look like button of your shirt. They are called circumvallate, and circumvallate papilla form inverted U or V shape in the back of the tongue, and the size is much larger, and only few circumvallate papilla are present in the tongue. 8, the number varies between 8 and 12. Okay, and each circumvallate papilla contains about 200 to 700 taste buds. So foliate a few hundred, buds in each per papilla. Per papilla, and circumvallate it ranges between 200 and 700 buds per papilla. Okay, and inside the taste buds you have the taste receptor cells. So first, if I go from large to small, tongue is the organ, right? Peripheral organ for gustation. Then on the tongue you have papilla, and you can see both the tongue and the papilla from outside, right? Then inside the papilla you have buds, and inside the taste bud you have taste receptor cells. Taste receptor cells, okay, TRC. And on the cilia or hair of taste receptor cells you have the receptors. It's like olfactory receptor cells. Receptors. Okay, so taste receptors are attached to the cilia. So that's from large to small. Okay. Now the location of the buds. If you see a fungiform papilla, this is a fungiform papilla, this one. So this is another, this is another. So these are the taste buds. So you see, in case of fungiform papilla, the taste buds are few that I mentioned in last slide, and they are located in the upper surface of the papilla. Okay, so located in the upper surface of the papilla, not um, at the edge or sides of the papilla. Okay, then if you see a foliate papilla, in case of foliate papilla, the receptors are not located in the upper surface. They're found in the sides of the papilla. So this is foliate. Circumvallate is also like that. Okay, so these are the birds. And circumvallate is much bigger, and same way in circumvallate we see the papilla are located not on the top or upper surface, but at the lateral sides, both sides. Okay, so this is how the taste buds are located. Now, if you take just one taste bud out from here and open it, it is like an orange. So you know that orange, although of course not that big, right? So this is a taste bud. Now inside the taste bud you have the taste receptor cells. So if you peel the skin of the orange and see inside, you'll see what? You'll see the receptor cells, and there are 4 different types of cells are found inside the taste bud. So these are the receptor cells and nucleus inside. So taste receptor cells. Okay, and at the apical surface or upper end you have tiny hair-like structures called hair or cilia. Let's say cilia. We used cilia for olfactory receptor cells too, remember, on the dendrites. So why for these three, first these three are for the 3 different tastes. Okay, so we have 3 taste receptor cells for 3 different tastes, and the last one here, this one is called the presynaptic cell. Okay, and these 3 are for 3 tastes, One is for sweet, one is for bitter, and one is for umami. So sweet, okay, bitter, and umami. So these three are for three different tastes. Why they are different? Because on the cilia you have the receptors, as I mentioned. So this is one of these three. So for example, this is for sweet. This is sweet taste receptor cell, and on the cilia you have the receptors for sweet chemicals like sucrose or glucose. Another one is for bitter. Another one is for umami. And the presynaptic cell is for other two tastes, salty and sour. So this one is for salty and sour. Both start with S. Okay. And the transduction occurs in presynaptic cell for salty and sour is different than the mechanism of transduction in sweet, bitter, and umami cells. Okay. In case of sweet, bitter, and umami, these first three, the transduction occurs. You know, transduction, we talked about that. Activation of the receptors and production of action potential. So transduction occurs for sweet, bitter, and umami cells by G protein coupling receptors. Okay, G protein mediated. So the receptor, this is the cell membrane. You remember in last lecture I talked about that. G protein has subunits, and the alpha subunit gets separated, right, from the G protein. And this is the receptor attached to the cell membrane, right? And on the inner side of the receptor you have the G protein attached. So when the receptor binds, the signal molecule binds with the receptor, then the G protein is activated and that alpha subunit is released, right, and activate the adenyl cyclase, and that way the sweet, bitter, and umami cells transduction occurs. And in case of presynaptic, salty and sour, these signal molecules directly open the ion channels. It doesn't, they don't go through second messenger or G protein activation. Directly get attached to the channels and open them. So these two are direct opening of the channels. So that's how inside the taste bud the cells are located. Okay, and if you see at the upper end of the taste bud you have a pore, and that is called taste pore. Okay, And what happens when you put the food in the mouth, salivary secretion occurs, and that food becomes like liquid, uh, and that is very helpful. The liquid would take the food molecules inside the taste bud through the pore. Dry food molecules cannot enter into the taste bud. However, after salivary secretion, the food gets mixed with the saliva, and that liquid pours into the taste bud through the taste pore. Okay, they must get in to get attached to the receptors on the cilia, right? And that would activate the taste receptor cells. Now, let me talk a little more about this taste receptor cells. So this is sweet cilia nucleus. Okay, so this is sweet taste receptor cell, and then this is for sour. Sorry, this is for the bitter. Okay, so this is bitter. And another one, this is for umami. Okay, so what happens on the cilia, you have the receptors, and these receptors are specific, and that makes the cells like sweet, sour, or bitter, or whatever. So these receptors are for sweet molecules. Okay, and here these are the signal molecules or food molecules here. Okay, so as I mentioned, these cells are activated by the second messenger, G protein mediated transduction occurs, and they release ATP. So they release what? ATP, all of them. Okay, release ATP. And these ATP are different than the ATP we get the energy from. These ATP have some kind of code inside them. Okay, and the ATP can do that. Do what? Can activate the nerve. These are the nerve fibers. Take the signal directly to the brain, towards the brain, and also this is the presynaptic cell for salty and sour. Presynaptic cell. Okay, and what happens? ATP, released ATP from these cells can directly activate the nerves, and then signal can go towards the brain, or ATP can activate the presynaptic cell. So ATP works in two ways. One is directly activating the nerve endings, or activating the presynaptic cells. Okay, and as I mentioned, these ATP encode the taste signal. Okay, so ATP released from this one encodes the sweet, ATP released from this one encodes the bitter, and ATP from this one encodes umami. Okay, and now presynaptic cells can be directly activated by salty or sour, okay, molecules, as well as can be activated by the encoded ATP. And here you have the synapse, the cranial nerve fiber, and the neurotransmitters are released at this synapse. So the main neurotransmitter that has been identified, that is released here, is serotonin. Serotonin. Okay. So you see that these are the cranial nerve fibers. Okay. Cranial nerve fibers will take the signal towards the brain. Okay, so that's how that action potential is transmitted to the brain. So ATP can directly stimulate the free nerve endings of cranial nerves, can directly stimulate, or can activate or stimulate the presynaptic cells, and then that will activate the cranial nerve. Serotonin, RO. Okay. So here in this picture you see the taste pore. Here these are cilia, or here different types of cells and cranial nerve fibers. Here you see the synapses. Now this is a kind of simplified way to show how the depolarization and action potential is produced in the taste receptor cells. So if you see here that this is G protein. Okay, these are the subunits, and sweet, bitter, as well as umami as I mentioned, they activate the G protein, and that would release the calcium from the endoplasmic reticulum. So intracellular, right, release of calcium. So this receptor is G protein coupled receptor. Okay, will be activated by sweet, bitter, umami. And these receptors would release the alpha subunit from here, the G protein, and that would activate the adenyl cyclase and will release the calcium from the endoplasmic reticulum. Okay, and this, so you see the calcium released here, and calcium will also enter from outside, from the extracellular fluid. And so the main thing here is the release of calcium internally from endoplasmic reticulum, or when that second messenger system will be activated, that will lead the calcium also get in through the calcium channels present in the bottom part of the taste receptor cells. So that the calcium will do what? Will rupture the vesicles that contain the neurotransmitter serotonin, will be released here at the synapse. Okay, and this is the nerve fiber. So action potential will be produced in the nerve fiber. Okay, and signal transduction occurs that way. And the ionic stimuli, salt and sour, those stimuli interact directly with ion channels to depolarize the taste cells. So here you see the salty and sour, these ionic chemicals or stimuli would directly open the ion channels. And both the receptors and ion channels are located, actually they didn't show here, in the apical end where you have the cilia attached. Okay, so sour and salty directly by opening the ion channels, and sweet, bitter, umami by transduction through the receptors. Okay, and when depolarization will occur due to influx of the chemicals, ions, that will open the sodium channel, potassium channel, as well as the calcium channel. Okay, so the main thing here is the release of calcium from the intercellular structure, as well as the influx of calcium. Calcium is important here to rupture the vesicles contain the neurotransmitter. Okay, Here you see I already explained this, three different types of receptor cells and ATP. It's not very clear here in this picture, and this is the presynaptic cell. Okay, and sour and salty would directly open the ion channels here and activate this presynaptic cell. Also, it can be activated by ATP that encodes the taste signal, three different taste signals. Now, on the cilia, as I mentioned, you have the receptors, and the receptors for sweet, bitter, and umami, the receptors are different. Okay, and for sour and salty, you don't need the receptors because they directly open the ion channels right here. So this is a cilia of presynaptic cell. Cilia, just one cilia of presynaptic cell, and you have the channels for sweet and sour. Okay, and there are two different types of sour channels here. You see here, this is one and this is another. Okay, so one is for sodium chloride, that we enjoy the salty taste, right? This is one type. Another type, this one is for adverse. High concentration salty signal or stimulus. Okay, so if you put a lot of salt on your tongue, then this one will be activated, and this is called adverse saltiness channel, and this one is like common sodium ion channel, low concentration. Okay, and another one is for sour. So three types of channels here, two for salty and one for sour. Salty channels, two types for normal concentration of sodium, and another is for very high concentration or adverse channels. Okay, and sour, only one type. So that is presynaptic cilia. Now if you see other three types of cells, the cilia, one receptor cell cilia has the receptors for sweet. So this is sweet. So if you see the structure of a sweet receptor, the sweet receptor has the combination of two compounds. One is called T1R2, another one is T1R3. So these two compounds or receptors pair together. Here for sweet, T1R2 and T1R3, these two receptors are paired together. Okay, so that is the structure of the receptor, these two, and this is the testant molecule, and the binding should be perfect here. Okay, so this is the sweet molecule, for example, glucose or sucrose, and this is the receptor. Okay, and for umami, this one, the receptor on the cilia, the structure of receptor is different. It is a pair of two receptors, T1R1 and T1R3. So only difference is here. Okay, and this one is same, and this is the testant and this is the receptor. Okay, and for bidder, you have very different. It's not a paired type receptor. It is a single receptor, T2R2. And as I told you, T2R2 and also T2R2, T2R, and it could be T2R2, T2R1 and T2R3. So it could be three different combinations, T2R1, T2R2 and T2R3. So three types. Here you can see receptors for beta. Okay, so you see that the shape of the receptors is different for different uh, types of receptors or taste receptors. They have different shapes and they are located in different cilia of different um, receptor cells. So now you know how the testant bind with the receptors on the cilia, and that will activate the test receptor cells. Depolarization will occur, right? And calcium would get in or will be released inside the cell, and that will rupture the vesicles, and that will produce action potential in the nerve fibers, cranial nerve fibers, okay, by the release of neurotransmitter. And now, then what happens? So from the papilla of the tongue, the taste receptor cells are activated, right, and signal first is taken to the brain stem. This picture is not very clear. Let me write it down. Brain stem. In the medulla oblongata of the brain stem, you have the structure called the nucleus of solitary tract. Brain stem has medulla oblongata, and in the medulla oblongata you have tractus solitaricus or nucleus of solitary tract. Same thing. Tractus solitaricus This is also called gustatory nucleus. Tractus solitaricus nucleus or solitary nucleus, which is the gustatory nucleus in the medulla oblongata of the brainstem. Okay, will receive the signal from the tongue. Okay, so this is the nucleus of the solitary tract here. Okay, and these are the cranial nerves. You see the taste signal mostly goes from the tongue, but also from, as I mentioned, pharynx, epiglottis. You have the receptors. Okay, so 3 cranial nerves here take the signal to the tractus solitaricus. From the front part of your tongue, the cranial nerve number 7. So number 7 is the facial nerve. Okay, number 7, cranial nerve number 7. The facial nerve. From which part? The front of the tongue. Front of the tongue. Okay, cranial nerve number 7, which is facial nerve, takes the signal to the tractus solitaricus. Here, this is the front of the tongue. And from the back part of the tongue, the cranial nerve number 9, glossopharyngeal. So back of the tongue, cranial nerve number 9, which is called glosso. Glosso means tongue, pharyngeal nerve. Okay, we take the signal to the tractus solitaricus, and from the epiglottis and the pharynx, wall of the pharynx, vagus nerve, cranial nerve number 10, from the epiglottis or pharynx, nerve number 10, which is the vagus nerve, will take the signal to the solitary nucleus. Okay, so here you see this is vagus number 10. This is glossopharyngeal number 9, and this is number 7, facial. Okay, and then the signal from the tractus solitaricus, signal is taken to the thalamus, which is the major sensory relay station. So of course thalamus will relay the signal. Then thalamus relays a number of sensory signals. So which part of the thalamus will process the gustatory signal? That is the ventral posterior medial nucleus. Ventral posterior medial. Okay, VPM, ventral posterior medial nucleus. Okay, and then from there the signal will go to the anteroinsular cortex and frontal operculum. Now these two structures are considered as the primary gustatory cortex. Okay, the primary gustatory cortex are these two structures here. Okay, this is frontal operculum in the frontal lobe. Both are actually in the frontal lobe, and this one is the insular cortex, anterior insular cortex. Okay, so from the thalamus, signal will go to these two structures of the brain. Okay, and these two structures, the neurons in these two structures produce the representation of the qualities of the testants: sweet, bitter, umami, salt, or sour. Okay, and help you to identify the testant. Makes sense? So what taste is that? So these two are the primary taste cortex. Okay, then the signal will go to the amygdala. This is the amygdala, and hypothalamus. So from the primary gustatory cortex, the signal will go to the amygdala, hypothalamus, and another structure. This is called the orbitofrontal cortex. You must remember these names from my last lecture. Amygdala is responsible for emotion, right? Hypothalamus regulates, in last class I mentioned that hypothalamus regulates the release of the hormones, right? Which occurs in the body when you smell. The hormones are released and that trigger many responses in the body. So in this case, hypothalamus plays a very important role to control the food and water intake. You know that hypothalamus controls food and water intake of food and water. So appetite. So in this case, this function of hypothalamus is important. Okay, in case of smell, release of hormone is more important. Here also release of hormone is important, but this is more important function performed by the hypothalamus here. Okay, and orbitofrontal cortex is very important. You remember the orbitofrontal cortex in case of olfaction too. The orbitofrontal cortex gives you the reward value of food. Okay, so reward value of food. Orbitofrontal cortex, and the orbitofrontal cortex gives you the reward value of the food, the enjoyment, okay, of the food. Same thing, in case of olfaction, the reward value of smell. So the perfume which one you like more and which one you enjoy more. Okay, so the two structures, frontal operculum and anterior cortex, they first receive the signal from the thalamus. So these are the first cortical areas to receive the taste signal, right? Primary taste cortex. Okay, now the flavor of food. You must remember, I mentioned at the very beginning that taste and flavor are two different things. So taste is a perception of sweet, salty, umami, sour, or bitter, uh, and these are the taste you get when you eat or drink something. However, the flavor is the total sensory experience. Okay, and a number of factors are involved in that total experience when you ingest some food. Okay, so the first thing is the basic taste, of course, that is the taste, sweet or sour or uh, bitter. You of course get that. Then the other attributes of testants that are also important: the pleasantness and intensity. So let's see here, taste, quality of taste: sweet, sour, salty, umami, sour, bitter, right? And you see here, this is a nice chart or cartoon. You see here sweet, salty, umami. These are pleasant. And then sour, mild sour is pleasant, but strong sour is not pleasant, unpleasant, right? And bitter is unpleasant. So those are the basic tastes and give you the pleasantness or unpleasantness. Okay. Another factor is the intensity. Okay, that is how sweet, how strong the sweet is, how mild the sweet is, right? And onset, after taste, those two things. After you put the food, you start to enjoy, right? Slowly. So that is the onset. And after you finish eating, that is also you continue to appreciate that. So those are factors how you enjoyed the food or how you are enjoying the food, right? Um, Localization, the tongue, where the food is getting attached to the receptors. That is another factor. However, so these all are related to the taste of the food. Then additional factors outside of the mouth that provides you the flavor of the food, help you to enjoy the flavor of the food. Olfaction from the nose, right? Smell, if the smell is good, you enjoy the food. If the smell is not good, you don't enjoy. Very closely related, right? Hunger or thirst. How hungry? When you are hungry, you enjoy the food more. When you are thirsty, you enjoy the drink more. Vision. How beautifully the food is being presented. That is also important. That's why we do the decoration, right? And presentation of food is very important. Visual signal. That also helps you to enjoy the food. Audition. Like, when you are eating, you will see some restaurants, they play music during, eating. So hearing or audition is also a factor that adds up. Okay, then mouth feel. A number of factors. Uh, the texture of the food, the temperature. We usually we enjoy the food after we warm it up, right? So before we eat, we warm the food. So warm food gives you better flavor and taste. Spiciness, the burning sensation helps. Actually, many people love that, right? That burning sensation also. That is somewhat a sensation, as you already know, right? Coolness, example mint, dryness. So those are uh, the factors. So all these are the factors you see. Not only the taste, many factors are involved to give you the complete, flavor of the food. So this is that flavor is the total sensory experience. Okay, evoked by the ingesting food. So that includes the basic tastes and many other things. Okay, so those are the things you need to know. Let's see anything we are missing. Adaptation of taste. Taste signal also has adaptation uh, like other sensory signals, and that's why, after you put the food in the mouth, you continuously move the food inside the mouth, and that is important. Otherwise the same molecules will stay in same location, and that will create adaptation, and you will gradually, adaptation, the sensation is lost or declines or diminishes gradually, so you don't want to do that, right? So that's why you move the tongue, you move the food on the tongue. So new food molecules will get attached to the receptors. Okay, and you will hear a term sometime that is called that super taster. Super tasters are the tasters who have a much higher number of fungiform papilla on the tongue. So if you see the tongue of a taster and a super taster, you will clearly be able to tell the difference here. You see here, this is the normal person, non-taster, and this is the tongue surface of a super taster. So you see the number of fungiform papilla is double or more than that in super taster. Okay. The term satiety. uh, indicates a reduction of appetite of food. Hypothalamus plays a very important uh, uh, role in this. Now, when you are hungry, you think that you will eat a lot. Uh, then you start eating, right? And gradually you lose the interest, and at one point you stop eating. So the hypothalamus plays a very important role, which is regulation of food and water intake. So you remember the signal goes to the hypothalamus. Anyway, so those are the things you need to know from the crustacean.